proceed looking on the process which is the glycolysis. And on the glycolysis, we have almost 3 stages, and here we are looking on the last stage, which is the oxidation by dehydrogenation. And we have seen that the result of this process, it will tend to form the 2 molecules of ATP from the 3 carbon compound. But also, as I promised you that we are going to start looking on this slide, that is when the 3 carbon compounds are separated from the phosphate group. So the 3 carbon compounds are converted to 2 molecules of pyruvate, each composed of 3 carbon, 3 oxygen, and 3 phosphate, and 3 hydrogen atom. So kama zile molecule za carbon sugar, hapa tukiongelea zile 3 molecules, you have to keep it into your mind that we are talking about the dihydroxyacetone phosphate and the glyceraldehyde 3 phosphate. So don't be surprised when I say 3 carbon sugar. So you have to keep it into your mind. So those 3 carbon compounds zikibi separated, meaning that the phosphate group now are tend to remove from those 3 carbon compounds. The result now, it will tend to form the 3 carbon compounds, which now will be converted to 2 molecules of pyruvate. So tukishaondoa phosphate group, tutapata molecule of pyruvate, ambayo kwenye hii molecule of pyruvate, each composed of 3 carbons and 3 oxygen together with the 3 hydrogen atom. So we tend to convert our molecule, which is a 3 carbon sugar. And when the glycolysis is complete, important products are produced, 2 molecules of NADH, 2 molecules of the pyruvate, which is the pyruvic acid, and 2 molecules of ATP. So tunasema kwamba ile glycolysis ikishaisha, sasa tumeshamaliza zile our process or our stages which are involved in the glycolysis. So our result now after the process of glycolysis, the first one we have seen that it is the 2 molecule of ATP. So tumepata 2 molecules of ATP, the NADH, and 2 molecules of pyruvate baada ya breaking down au baada ya kuondoa phosphate group kutoka kwenye ile 3 carbon sugar. And also tukapata 2 molecules of ATP. So this one is just our result of the completion of the first stage of aerobic respiration, which is the glycolysis. So if you can be asked into your exam, maybe mention the result of the glycolysis or mention the effects of the glycolysis. We have seen that we have got 2 molecules of NADH, 2 molecules of ATP, and 2 molecules of pyruvic acid or pyruvate. So the NADH and pyruvate are used into the next stage of cellular respiration, and the ATP molecule are used for the reaction in the cell that require the energy. So hile NAD pamoja na hile pyruvate ambazo sasa tulishapata kutoka kwenye ile process ya glycolysis, tumesema kwamba hizi molecule mbili zitaendelea na next stage of cellular respiration, kwa maana kwamba itaingia moja kwa moja kwenye hile Krebs cycle pamoja na hile electron transport chain. Tutaenda kuangalia kwa upana zaidi tukifika kwenye hiyo part. Lakini pia hapa tumesema kwamba ile ATP iliyozalishwa kutoka kwenye glycolysis itasaidia kwenye masuala mazima ya ile first stage of glycolysis. You know that the first stage of glycolysis you tend to use ATP. So hii ATP ambayo imetengenezwa hapa ndio ambayo itaenda kutumika kwenye ile first stage of the glycolysis. So ile kwa sababu ile stage inahitaji presence of energy. So that energy will be used on the first stage of a glycolysis. And also the process can be detailed as follows. That is, the glyceraldehyde 3-phosphate or 3-phosphoglyceraldehyde, that is 3-PGA, is converted to 1,3 biphosphoglycerate or 1,3 diphosphoglyceric acid, which is 1,3 DPGA by glyceraldehyde 3 phosphate by this enzyme, which is the dehydrogenase enzyme, whereby the NAD is reduced to NADH. So hii ndio process nzima inayotokea mpaka ile NADH inapatikana kwenye ile process nzima ya glycolysis. Kwa unaweza ukaona kwamba hapa ile 3 carbon sugars, kule mwanzo tulikuwa tunasema 23 carbon sugars, tunaiconvert mpaka tunakuja tunapata ile NADH. So hapa ndipo tunasema kwamba hii molecule 3 carbon sugar ambayo tunaita glyceraldehyde 3 phosphate tunaiconvert kuja kupata biphosphoglycerate. So this biphosphoglycerate ambayo tunaita 1,3 diphosphoglycerate by huyu enzyme ambaye inaitwa glyceraldehyde 3 phosphate dehydrogenase enzyme, meaning that huyu enzyme ndiye ambaye ataconvert, ataifanya hii reaction au atacontrol hii reaction mpaka kuja kupata 1,3 diphosphoglyceraldehyde. And then our molecule, which is the 1,3 diphosphoglyceraldehyde, now tena inafanyiwa conversion, that is it is converted to 3 phosphoglycerate or 3 phosphoglyceric acid, which is the 3-PGA by phosphoglycerate kinase enzyme. So this process is involved the loss of phosphate group, which is the PI, from the this molecule in order to form the 3-PGA, and the PI now is transferred directly, or that molecule it is transferred to a molecule of ADP to form the first molecule of ATP. So this is what is happening. So ATP inatengenezwa kutokea huku, kwa maana kwamba tukiconvert hii our molecule that is 1,3 diphosphate, ndipo ambapo tutapata hii molecule ambayo inaitwa 3-PGA. So hii 3-PGA under the conversion, meaning that we tend to remove the phosphate group. Remember here we have di, meaning that we have 2 molecules of phosphate group. So we tend to remove a phosphate group from this molecule. So tutaiondoa hii phosphate group, ndipo ambapo tutapata 3 phosphate group. So kama tumeondoa phosphate group, hii phosphate group na ile ADP ambayo ilitengenezwa kwenye the first stage, ndipo ambapo itaungana, that is PI together with this ADP, tutaenda kutengeneza our first molecule of ATP. So unaweza ukaona, and actually 2 molecules of ATP are formed because there were 2 triose produced in the lysis of a fructose, that is 1,6 biphosphate. This is what I was explained earlier. That is his molecule mbili za phosphate au molecules mbili za ATP zinatengenezwa kwa sababu tu ile fructose molecule baada ya lysis yake tumesema tunapata 3 carbon sugars, and the first carbon sugar we have said that it is just glyceraldehyde 3 phosphate and dehydroxyacetone, and in order for the dehydroxyacetone to enter into the glycolytic pathway so that ATP can be formed, it must be isomerized. So baada ya kufanyiwa isomerization ile dehydroxyacetone, ndipo ambapo sasa tutaenda kupata ile molecule ya glyceraldehyde. So ile glyceraldehyde 3 phosphate itapitia kwenye zile stage kama kawaida, itaenda kuform ATP nyingine, molecule nyingine ya ATP. So but the 2 ATP formed are used to pay back the initial ATP used during the first process, which is the glycolysis, which is phosphorylation of a glucose stage. So unaweza ukaona kwa maana kwamba hii ATP iliyozalishwa inaenda kurudishwa kule nyuma. Tunasema kwamba ni kama inalipa ile ATP iliyotumika kuivunja ile glucose molecule kuja kupata glucose 6 phosphate. Kumbuka kwenye first stage of glycolysis ilikuwa inaitwa phosphorylation of a glucose stage, we tend to use ATP. So where does that ATP come from? It comes from after now formation of ATP in the process of glycolysis. But also the formed 3-PGA, now it is converted to phosphoglycerate or 2-phosphoglyceric acid, which is the 2-PGA by the phosphoglyceromutase, which is the phosphoglycerate mutase enzyme. So hile PGA ambayo tulipata pale pia inafanywa conversion ili kupata hile 2 molecule ya PGA under the enzyme. So the enzyme ata-rearrange hile phosphate group from the third carbon, which is the 3-PGA, to the second carbon, hence forming 2 phosphate group, which is the 2-PGA. So the 2-phosphoglycerate, which is a 2-PGA now, is then converted to form the phosphoenolpyruvate, which is the PEP, under the influence of the enolase enzyme. And this reaction, water molecule is removed from to form 2-phosphate glycerate. So hapa tunahitaji kutengeneza pyruvate. So ile pyruvate inatokea wapi? Ndio inatokea huku kuanzia kutokea kwenye ile phosphate molecule, which is the 2-phosphoglycerate molecule, inaenda kuform PEP. So then the phosphoenolpyruvate, which is PEP, then it is converted back to form the pyruvate. But this reaction, it will be under the enzyme ambayo inaitwa kinase enzyme, whereby the phosphate group, which is the PI, which is a PEP now, is transferred to ADP molecule and combined to form the ATP molecule. So unaweza ukaona kwamba ATP molecule inazalishwaje. So again, 2 molecules of ATP and 2 pyruvate formed as explained earlier, because the triose phosphate has to enter twice the glycolysis processes. That is, when the 3-PGA is used, the dihydroxyacetone phosphate is converted to 3-PGAL and enters to the cycle. So the formation of the pyruvate and the glycolysis process. So we're going to see this in the figure 6.9. And this figure, my student here, I promise you we're going to see later on to the next session. Tutaenda kuangalia summarize the question or summarize figure of formation of the PGA, that is formation of the pyruvate, ili endelee na process ya Krebs cycle. And by saying so, this marks the end of our today's session. So thank you for following us, Educare TZ. Bye bye, have a nice time.